Okay, in the second part of chapter 13, we are going to talk about how DNA is replicated, and then we'll just really briefly at the end of this lecture talk about how mistakes or damage to DNA could be repaired. All right, so hopefully you remember when we talked about the cell cycle a couple of chapters ago that DNA replication occurs during S phase of the cell cycle, and this is going to happen before any kind of cell division, mitosis or meiosis, can occur. We've got to copy all of the DNA before we carry out any kind of division. Um, and in the first half of chapter 13, I ended by saying pretty early on, right after the discovery of the structure of DNA and the fact that DNA replicated in this semi-conservative manner, um, pretty soon thereafter, a researcher found that DNA polymerase was kind of the key enzyme involved with copying the DNA, with building new DNA molecules. It's an enzyme in cells. And as other researchers dug into this process, they found, yes, that is true. DNA replication is critical, but it turns out there's actually a whole handful of other proteins, most of them also enzymes, that are playing important roles as well. So what I'm going to try to do in this lecture is kind of walk you through that process and what some of these different protein enzymes are doing during DNA replication. And I think it's a little bit hard to get across in static images, so I'm going to post a link to a YouTube video on Canvas that would show these things happening in motion. And so if you're not feeling it from the PowerPoint here, I definitely recommend go take a look at the video afterwards. So this whole process of DNA replication has to have some start point, right? We've got our big long DNA molecule, two strands of DNA, and if you remember the semi-conservative model for replication, the first thing that would happen is we separate the two strands. And it turns out it's not really feasible to just completely unzip the two strands of DNA, along the whole length of the chromosome. They would just float apart from each other, um, and it would be a bit of a mess. So what will happen is the cell will just unzip a small portion at a time, and this will start at a site that's called the origin of replication, which makes sense, right? Where are you going to start replication? At the origin. Um, and usually the first thing that will happen is we will unzip a portion of the DNA. And then, as we're going to see before DNA polymerase can start adding any new nucleotides, building any new DNA, it needs what's called a primer. And this is because DNA polymerase can only add new nucleotides onto a 3' end of an existing polynucleotide. And if you look here, for example, imagine this is a piece of unzipped DNA. Well, where's the 3' end that DNA polymerase could add onto? It's way out here at the end of the chromosome, and that's not where we want to build the new DNA, right? We want to fill in this region here. So to get around that, cells have an enzyme called primase that will come in and bind to the DNA, and it will build a complementary, what's called a primer, which is just a short stretch of RNA that's complementary to the DNA. And because RNA is built of nucleotides almost identical to DNA, right? The only difference is there's uracil instead of thymine. The RNA can actually hydrogen bond with the DNA. So we get this little RNA primer, and there would be right here, there would be a 3' end of that RNA primer, and now DNA polymerase could start adding DNA nucleotides, onto that little RNA primer. So primase would come in first, lay down this primer, then DNA polymerase would come in and bind and start adding DNA onto the end of that primer. So if we look down here at the bottom, all the red here would be newly built DNA. There's different versions of DNA polymerase depending on whether you're talking about eukaryotes or prokaryotes, but I'm just going to keep it generic for our purposes, and we're just going to call it DNA polymerase. The enzyme that actually unzipped the DNA to start with is this one called DNA helicase, and you could see DNA helicase down here in red. So it would actually bind to the DNA and move away from that origin of replication, sort of unzipping as it goes. And so you could imagine there's one helicase here moving off to the right, unzipping the DNA, and it's not shown, but there would be another one over here at the left side of the screen, moving off to the left, unzipping in that direction. So this, what's called the replication bubble here, would be this region with the DNA strands that have been separated. That replication bubble is constantly growing and moving towards the ends of the chromosomes. And have a little bit of a problem as helicase comes in and starts to unzip. What would happen is, because there's complementary bases here on both strands of DNA, they want to stick back together and hydrogen bond with each other, and they would just immediately do that if it weren't for these single-stranded binding proteins, little blue circles here. So these come in and sort of coat the single naked strands of DNA just to physically prevent them from sticking back together right away. And then another enzyme is working out here, sort of ahead of what's called the replication fork, right? This would be the fork in the DNA here, called topoisomerase. And what topoisomerase does is it's just going to relieve tension that's going to build up here as we unzip these two DNA strands and prevent the DNA from breaking. You could, if topoisomerase wasn't here, you would actually get so much tension out here and on the other side of the fork that you would get breaks in the DNA, which would obviously not be a good thing. Topoisomerase intentionally will make little nicks in the DNA, let it unwind a little bit to relieve some tension, and then seal it back up again. And you can see, if we look in the middle of the fork here, you know, as we first unzip these two strands, polymerase came in, and you can see it laid down a primer here in the green on the top, complementary to the top strand, and it laid down a primer here in green, complementary to the bottom strand. And as we just discussed on the last slide, after that primer's made, DNA polymerase comes in and will bind to both of these primers. So when it binds to the primer on the top, it starts adding DNA nucleotides to the 3' end, and it's just going to move off to the right, reading that template DNA base for base as it goes. Okay, here's a G, let's pair it with a C. Here's a T, let's pair it with an A, all the way down the line. And it'll just keep going, right? It's basically just going to follow along behind that helicase as far as it goes, maybe even to the end of the chromosome, assuming it makes it that far. On the bottom strand here, you'll see what will happen is, instead of this long continuous synthesis, you're going to get DNA synthesized in these little short fragments that are called Okazaki fragments, after the researcher that discovered them. And you can see here, so the original primer that was laid down here, DNA polymerase would move off to the left. We don't see it working over here, but there's presumably a DNA polymerase molecule out here off the screen on the left that's still moving, just like this one up here, just in this long continuous synthesis. But as we unzip the DNA a little bit further, we were left with a gap here now to the right of this original primer. There was nothing. And so essentially, as soon as there was enough room, primase enzyme would come in and lay down another primer. And then DNA polymerase would bind to that and extend it back until it bumps into the previous primer, and then it falls off. We unzip the DNA a little bit more, and primase jumps on down here again and lays down another little primer, and then DNA polymerase will come in shortly and extend that back to the previous primer. So we get this distinction here where, one side of the replication bubble is just continuous synthesis. It's called the leading strand. And then the other side of the replication bubble is going to be these short Okazaki fragments, sometimes also called the lagging strand. But again, this is just proceeding either until we hit the end of the chromosome, or maybe we bump into another replication bubble coming from, the other direction. And so, yeah, here's just another illustration of what that might look like, right? You've got continuous leading strand synthesis up here, just one long new piece of DNA, right? It keeps getting longer and longer, but it's all continuous. Whereas on the other strand, we extend that original primer. We're left with this blank space here now. Primase comes in, lays down another primer. We extend that back until we bump into the previous, and so on. Now we still have a little bit of an issue. We do that process along the length of the whole chromosome until we've got all this new DNA made. But we're still left with these RNA primers, right, throughout our chromosomes, and we don't want the RNA there, right? We want our DNA molecules to be all DNA. So we've got to get rid of those RNA primers before we're done with the replication process. And a DNA polymerase molecule will come in and actually remove the primers and then fill in the little gap that's left. And it can do that because there is now a 3' end here that it can extend off of to fill in that gap. And once the gap's filled in by DNA polymerase, yet another enzyme comes in, DNA ligase. If you ligate something, you're basically gluing it together. So the job of DNA ligase is to just seal these tiny little gaps between nucleotides there so that it's all just smooth and continuous. So after that's all done, we've removed all the primers, right? We've sealed up all these little nicks in the DNA, and then we should truly have, completed that replication process. We've got, one original strand, one newly synthesized strand of DNA stuck together. That'll be that semi-conservative replication. And so you see all these enzymes coming and going, and it might seem like, gosh, it's going to take 10 years to replicate the 3 billion nucleotides in our genome, but it actually happens amazingly quickly, and in part that's because DNA polymerases are what are called processive, meaning they can catalyze a lot of linkages every time they bind to DNA. So they're working at a breakneck pace. And there's other proteins like this sliding DNA clamp that can sort of help keep the DNA polymerase centered and focused and lined up just perfectly on the DNA, so it's not gonna fall off. And I wanted to include this table for you just because a lot of the questions I tend to ask regarding this material are just basic, what are the functions of some of these different proteins that we just went over. So for example, helicase unwinding DNA, or topoisomerase relieving some tension, etc. So I think this table is just a nice sort of summary to help you study the jobs of those important proteins. And we started all this talking about this idea of an origin of replication, and I wanted to come back to that and just make the point that in prokaryotes that usually have very small chromosomes and usually just have one circular chromosome in most prokaryotic cells, bacteria for example, those organisms just tend to have one origin of replication. So on that circular DNA molecule, there would just be one point where this replication bubble starts, and then the bubble would proceed off in both directions until it had gone all the way around the circle, and we'd replicated the whole chromosome. In eukaryotes, like our own cells, because we have very large linear chromosomes, right, 46 linear chromosomes in each one of our cells, it would just be too inefficient to try to replicate the whole deal with just one origin of replication per chromosome. So every one of our chromosomes has hundreds of origins of replication. So you've got situations like this where you've got multiple replication bubbles right next to each other, getting bigger and bigger, and eventually they all kind of run together, and we've completed the whole process of replication. So that also allows for the S phase to be pretty quick. Another thing to point out, and this is really interesting to talk about, is there is still one little issue with this replication process at the very ends of chromosomes. So if we were to have followed that process, all the way through to the end of the chromosome, we would have seen on one of the two strands there was an RNA primer right there at the very end of the chromosome, and DNA polymerase can come in and remove that, but there is no 3' end available to allow this gap to be filled in, right? We do have this 3' end up here, but we could only extend that out this way to the right. We can't really fill this in down here. And so what happens is every time one of your cells goes through a round of DNA replication, each one of your chromosomes gets shortened at each end by the length of one RNA primer. And it's, generally speaking, not a problem, because your cells know this is going to happen, and they've actually accounted for that fact. The ends of your chromosomes just have this just repetitive sort of garbage sequence of DNA that are called the telomere sequences. So it's just, like the same DNA sequence just repeated over and over again. There's no genes, no real important information there. And those telomeres are basically there just as a buffer, right? Your cells know, okay, those telomeres are going to gradually get chewed away over time as your cells divide. But if you have cells that divide enough times, what will happen is you'll actually chew through the whole telomere, and then that sets off alarm bells that, hey, we're in dangerous territory here. We're going to start chewing away at DNA that contains genes and really important information, and that will trigger apoptosis, which was that programmed cell death. So this is one of the reasons, as you get older, you always hear elderly people say, "Oh, I feel like I'm falling apart," and to some extent, literally, you are, because your telomeres are getting shorter and shorter the older you get, and at some point, your cells start going through apoptosis, and you're literally losing cells and tissue left and right, which contributes to aging in a big way. And so there's a lot of researchers that are working on this, trying to understand how this process works and see if it could be slowed down or even reversed. And I think they are making some headway in that regard. So we may have some real treatments for aging that tie in with this whole idea of telomere shortening. And one potential treatment, and this is actually part of all of our biology, is that our genome does contain a gene for this enzyme called telomerase, which will actually regrow telomeres. And usually this enzyme, or this gene, only gets expressed, this enzyme only gets produced in like adult stem cells that have to divide, basically your entire life, right? For example, you have stem cells in your bone marrow. I think we talked about that in a previous chapter, and those are dividing throughout your whole life. And because they're going through so many rounds of cell division, they would tear through their telomeres really quickly, much faster than the rest of your cells. So they solve that problem by kicking on this telomerase enzyme and just regrowing the telomeres, so that they don't have that issue. So, that's one thing researchers are interested in as well. Maybe we could, turn on telomerase in other cells and regrow some telomeres there. But the big but there is, researchers have also found that this telomerase enzyme is almost always turned on in cancer cells, and that's one of the reasons cancer cells are often immortal and can divide indefinitely. So you sort of run this fine line, right? Yeah, it would be great to reverse aging using telomerase, but we also don't want to give ourselves cancer. But again, like I said, there's a lot of interesting work going on there. So lastly, I just want to talk really quickly about how errors in DNA sequence or structure could be repaired. And there's really these three main repair mechanisms. There's proofreading, there's mismatch repair, and what's called excision repair. And 2 and 3 are really just sort of variations on the same theme. Um, but proofreading is just like it sounds, is referring to the fact that the DNA polymerase enzyme can actually proofread itself. So, as DNA polymerase is going along the template DNA strand and building the new complement, it literally double checks itself to make sure it's inserted the right base. So in this case, it didn't. Wrong base came in and momentarily paired or tried to pair, and DNA polymerase turned on its spell check and said, nope, that's not right, and kicks it out and waits for the correct one to come in. So DNA polymerase is very fast, and it's also very accurate, but that's not to say it doesn't ever make mistakes. Sometimes it does, and it misses them occasionally. So you will get some level of mistakes there. So if DNA polymerase was to insert the wrong base, you would end up with what's called a DNA mismatch, right? Where you have this nucleotide pair here that should not be paired together. We should have, maybe this was a G down here, we should have inserted a C up top, but we put in a T instead. So we've got that mistake. Well, there is a mismatch repair mechanism, which is basically an enzyme that will come in and identify the mismatch, cut it out, so you're left with a little gap, and then DNA polymerase can come in and fill in the gap and hopefully not make the same mistake again, and fix that. So proofreading, mismatch repair, and then lastly, we have excision repair, which, as I said before, it really looks a lot like mismatch repair, but it's for, instead of mismatches, sort of slightly larger scale structural issues. For example, if you are exposed to a lot of UV radiation, just in form of sunlight, it will cause these weird bonds to form between adjacent thymines in your DNA, and you'll get what's called a dimer, which causes like a weird little bulge in one strand of the DNA and can cause problems and lead to things like skin cancer down the road. But again, you do have this excision repair pathway that should recognize some of those kinds of issues. For example, if this was a dimer, it would see that, cut it out, and then DNA polymerase could fill it in. So I know it's kind of reassuring that you've got these mechanisms in place to try to keep your DNA healthy and having the correct, quote unquote, correct sequence. All right, that is it.